Um, you know, you know what my personal nightmare would be? Being microbiologically educated, then watching my genome sequence and being like, "What the fuck is this shit?" Well, that's what we're gonna do tonight. Um, but let me search quick. Let me Google search quick. High throughput sequencing, and we can quick look at how this is obtained. It's conceptually not that bad. So I can just find a good image. We can talk about it super quick. It is very clever. Wait, what if I just pull up the lecture I gave yesterday? Oh, make a new record or something command so you can remind me. I need everyone to remind me to record. Oh, record. Yeah, I'll do something like that. Seriously though, is that a realistic perspective? Could you just read it out like that? Like read it out like this? Like this is real. Hello, Alt Miss Frizzle. What do we have here? We have my DNA, specifically my mitochondrial DNA, which looks a little suspicious. All of the colors mean that it's a variant. I believe that just kind of means that something's wrong with this region. Uh, Here, I know. Wait, hold on. MC1R. Let's go to MC1R. So down here, yeah, it is sussy DNA. Oh, my mouse doesn't show in this view. That's a little annoying. Actually, hold on. Can I change that, please? Properties, show mouse, capture cursor. There we go. Or not. Oh, there we go. Okay. So this right here is a gene, specifically the MC1R gene. Does anyone know what that does, or why I might know it without looking it up? You meant reading out like, oh, damn it, I'm susceptible to cancer. Oh, damn it, my liver won't work the way it's supposed to, etc. Kinda? Okay, David, right here, right here. I just happen to know this gene off the top of my head. If you look, this is the gene body, this part with the arrows. So if we zoom in a bit more, actually a bit more, you'll see the sequence itself. So all proteins start with a methionine. So it's the start codon, goes. And above it, you can see all these little gray bars. Those are individual reads. So if I double click on one, this is this particular read, 150. Oh, you can't see this little window. That's super cool. I might change up how I'm looking at this. Anyways, it's an individual read, so 150 bases of DNA that was read by a machine. And then it's not just that 150 bases, it's millions and millions of them being read, so that in aggregate, they all pile up like this, with the average being shown up here on the top. Not the average, the pile up. Just a representation of all these individual ones, all in one continuous line. But you might notice a little red dash here. So this little red dash is representing a place in my genome where I differ from the reference right here. So the reference in this is a C in this position. So C is the most common, if I remember correctly, but it is a mutation. Yes, Brian. This particular mutation, when you have two, I know I have two because, well, I'll tell you in a second, but I know that I have two genetically because 100% of the strands of DNA that were sequenced here show the same thing. So it's very likely that I sampled, both homologous chromosomes equally, from one from mom, one from dad. This T gives me my red hair. So this is the particular specific mutation, but really SNP, single nucleotide polymorphism, which is just any difference that can show up in a population. But this one right here is what gives me my red hair. So that's a very, yes, this one alone, this right here. Now it doesn't quite explain like the color, like why my like eyebrows are a little bit darker. that has to do with sort of the expression of other genes potentially. But in terms of red hair pigment, it is this gene specifically right here. So that's pretty cool, right? That's like the easiest one. All right. Let me see. I'm just gonna change up really quick how we're viewing this. How am I gonna do that? Give me 1 second. Display capture? Is that what I want? Yeah. Uh-oh. Uh-oh. That wasn't good. We're going to display 2. There we go. We're going to look at that. Okay, we're going to put it down here. And we're going to quiet these down. I just have to change this a little. Sorry, I'm not going to be paying attention for the next couple seconds, just so I can kind of get this all figured out. There we go. That's a little bit better. So now you should be able to see this, and if I click on something, yeah, you can see the reads. So individual reads have names and a bunch of information about them. Okay, so you got to ask now, when it comes to a phenotype like hair color, do you as a brown-haired fellow share the same genes with any other brown-haired person, or are there various combinations that will achieve the same result? That's a good question. So there's fairly simple traits, but those simple traits, so for example, I'm pretty sure, if I remember correctly, blue eyes in Northern Europeans are probably from one individual. So like one ancestor tens of thousands of years ago had blue eyes, and that's why all Europeans who have blue eyes are related to this one person, but super long time ago. So long ago it doesn't matter. There are other blue-eyed mutations in other parts of the world that are completely separate. So to answer your question, it's both. It depends on the particular mutation. So for example, anyone in my family who has red hair probably shares literally the same gene mutation, this one, from the same original chromosome, one of our ancestors. That said, I don't know if red hair specifically has more alleles, because there's certainly other shades of red hair, and I think that has to do with other genes, so combinations of other genes do play into it. And that's for fairly simple phenotypes. Other things like height are super complicated. Many, many, many genes contribute to height. So you have to get the same, so if you and some other person are the exact same height, there is no way, even if you're related, no way you have the exact same sets of the many genes as each other. It just so happens that whatever sets of alleles you do have combine in a way to give you the same height. Human exposes his own DNA. But are there other genetic combos that would provide red hair as well? Probably not the exact same shade, but in general. I don't know that for sure. I think this is the red hair gene, and then that's modified by other things. So for example, my sister has strawberry blonde hair, so red hair with blonde streaks, but I don't know why. Yeah, I don't know why in particular. %, of redheads have MC1R gene variant. Thank you, Brian. So that's the answer right there. So it sounds like the other 20 have something else, which shouldn't be surprising. Okay. Hold on. I was opening a PowerPoint. Let's see. PowerPoint. Okay. So, PowerPoint. So the way they did this was through something called next generation sequencing. It is massively parallel, so you are sequencing hundreds of millions of fragments all at once. And what you do is you basically get your DNA sample and fragment it. You can fragment it using a number of techniques. You add on adapters, which are just short sequences, and you can add that enzymatically. So an enzyme does it for you, just sticks them right on there. So the unknowns are these red, green, blue. The known, though, are these light gray and black. So we know those sequences. You attach it to a flow cell, which is effectively a special little cassette thing, glass slide with more of these little bits of DNA chemically bonded to them. So through something called base pairing, these fragments will attach to the flow cell. It's just that, A sees a T and G sees a C, and they hang out together. So they get stuck, and then they just kind of amplify. Not going to talk about that, but those amplifications occur in such a frequency that they form these little clusters. And then they go through the actual sequencing reaction, where going down this bit of DNA, they just add new bases as if they were copying that DNA. So A always goes with T, T always goes with A, C always goes with G, and G always goes with C. Now these little circles right here represent individual DNA bases being added in order, but also they are fluorescent. So they have these little colored chemical bits on them that a camera literally looks down at. And so it's watching each one of these little clusters. And as you're adding these in, you tell what the colors are, and it tells you what base it is. So this takes a series of pictures. And so this is 4 steps right here. So it's just showing that for this one, it would see a T, and then second step, it would see an A, second or third G, fourth C, and so on and so forth. In my case, 151 times per what is called a read. Each one of these is called a read because you're reading the DNA. So each cycle determines one base at a time, but each spot is tracked for 50 to 150 cycles. And this is a real image from one of these sequencers where each one of these little pretty dots is one cluster of DNA representing one fragment of DNA. And the different colors represent the different bases that are being added. So yeah, that's the short and sweet of it. Oh, Miss Frizzle, you have to, uh-oh. Oh no. Is that not there? Why didn't that work? I'll try to fix that in a second, David. Yeah, no. I can commiserate with never being prepared. Well, not never. I shouldn't say you're never prepared. I'm never prepared for class, though. At least not when I want to be. Okay. Let's, uh-oh. Oh, the community challenge has ended. Oh my God. I have to get some more packs of cards. Yeah, so what did I say with that? So the next non-guest stream will open some cards. That's going to be Sunday then. Because hopefully I'll have a guest to talk about alien chromosomes and not replication, reproduction on next Wednesday. Okay, let me try that again. Trek. Oh, there we go. I don't know why it didn't work. There you go. Fix it. Well, thank you, Miss Frizzle, Alt Miss Frizzle, for contributing the last necessary points to that. I will have to remind myself of that So I have some fun Voyager cards to open up. Maybe I'll go to the flea market and grab some more cards for 25 cents each per pack. Okay. You're still trying to get through cell transport. They wouldn't even tell you what diffusion across water was called when it was right in front of them on the graphic organizer. Can't think about next week. Yeah. Yeah, it's funny because I have someone I work with who's teaching intro bio for college right now. They're also on cell transport right now. That's something that I struggled with my students with, and I was telling her that she's gonna struggle with it too. Not for any, reason that was her fault. It's just students sometimes don't really care about stuff transporting in cells, which is sad. Whoopsies. I'm just moving some stuff around now. I want to make sure that with this new window we get maximum visibility. All right. Yeah, I do mean Star Trek Voyager. They did have cards, which is I was surprised by it, which is why I bought them. Okay, so what else do we have is kind of the question. Whoa, don't look at that. Actually, you can look at this. I'll just do that. So I got a bunch of results. So the thing we're looking at is called a BAM file. It is pretty big. So it is a binary compressed file. And so that is pretty small, and yet it's still 10 gigabytes. We have some raw data. This is only a small, like 2% of the raw data ended up being 11 gigabytes before I stopped it decompressing because I was going to start to stream. And then we have some insertions, deletions, SNPs, and then kind of just everything. So we can look at those too. You want to look at PUM 1 and 2? I can do that. You've been doing Osmosis Egg Lab for a week. You've known about osmosis and tonicity since you took notes on it last week. Yeah, well, that requires remembering. Alt Miss Frizzle. Oh, Brian, PUM is big. Is there anything in particular you wanted me to look for? Skimming already. It looks like I have a couple intron variants here and there. Ooh, also one thing I have to do when I realign this myself, they said it's 30x coverage. This does not look like 30x coverage to me. 30x coverage just basically means that every single base on average has 30 sequenced bits to it, like sequenced reads, and that does not look like any part of my genome. Yeah, it's not 30x. So I'm definitely gonna like email and be like, hey, I'm literally a genome scientist. I know that it's not 30x. Sequence more or give me my money back, please. Hey, oh, here's one. Okay, so, oh, that's very low coverage though. I don't think I trust that necessarily. Yeah, this is low coverage too. I mean, like, what are the chances that that's actually correct? Little tiny UTR stuck on there. You told them their grades are gonna look like you soak them in a hypertonic solution if they didn't get it together. Yeah, you called them out, and I will call them out. You're just curious since you study PUM variants, yes. Oh, zoom in on that 5' UTR. You got it. Itty bitty human 5' UTR. Do I actually have a G there? Maybe not. What are we looking for? Reverse the strand sequence. Sure can. Is anything popping out yet? ATG. Oh. What? Am I doomed? What's cool? Also, like a read depth of 2 isn't much. So that could, hold on. Map Q quality. So I'm checking the quality of this read. It's pretty good actually. Base equals G at, is that what I'm looking at? Wait, am I looking at the right thing? Whoops. Come on. So, puff proteins bind their own 3' UTRs to regulate them, and you have a possible mutation in the binding site. Oh, 5' UTRs. Really? Should I be called in for observation? I guess I should know that because I studied them for a time. But I forgot what the sequence was. Show me the read. Show me the read. Yeah, okay, so that was very well mapped. I mean, could be cool, but I don't actually know. Let's see. Well, let's check the other part of the 5' UTR. Okay, there's more coverage, but it's see, I mean, like, look at this. It looks like I have the same number of mutations here, but it's clearly a sequencing mistake since all the other reads are showing the canonical sequence. So let's just say that's that's with a grain of salt. You also wanted PUM2, you said? PUM2. PUM2. So you're saying you are a practically minded guy. What can I actually do with all these results? See, that's the thing. Not a whole lot. So you can look at some like known results and stuff like that. Known gene variants, like the red-haired thing. There's some other that are suggested to be related to certain diseases, and I'll show you how we can do that. But honestly, this was more about curiosity and to say that I've done it. I also do want to go and look around for like deletions and insertions, kind of like bigger problems, because if you look, like I said, any one of these like dashed colored lines, that's a variant. There's variants all over the place. But most of them are inconsequential for my life. So let's see. Yeah, so this one has a ton of introns as well. Not really. Oh, this might be a variant that's. Oh. Okay, so that's a little bit more. Wait, is this the UTR again? We should look at my CCR5 gene. That's a good. Yes. So if you don't remember, we talked about this gene, CCR5. Is it CCR5AS? CCR5 is a gene that there is a 30-some base pair deletion, and in that 30-some base pair deletion that causes you to be less susceptible to HIV. Yes, that is the one that provides immunity to HIV. So this is the practical part in sort of telling us whether or not, possibly I have resistance to a certain disease. Okay, so it looks like there's a couple. Expand. There we go. There we go. Okay, question is, where is the deletion supposed to be? Oh, and also, so another problem is just how they sort of aligned these. There is a chance that they are not showing the reads we would have to see in order to determine whether or not I have a particular deletion or whatever. So when the program is going through and saying like, oh, this thing right here, this read definitely sits here, it's looking for matching. And if there's too many mismatches, it throws it out. So if there's a big deletion in the center, there's a chance that it's thrown out. So this looks like a deletion. So 3 base pair deletion right here, but it's in a super repetitive region. So I almost doubt that that's even real. Okay, we gotta look up exactly though what the CCR5 gene is. Not what it is, but what the deletion is. CCR5 HIV. It's delta 32. I remember that. Delta 32. Like, what are the coordinates of that? Oh, that's what I should do. Coordinates. Okay, 32 base pair deletion in the open reading frame. So that is this big blue block down here. That's the thing that actually makes the gene. Can I make it change? Change track color? Oh, that'll just turn it, whatever. Whatever. You hate remember all this. You gave up on COVID. It's BJ1. Sounds like something you read on a red light district receipt. Sort of does. All right, but where is this mutation? Or where's this deletion supposed to be? Oh, wait. Actually, so back to this. There is what should be a list of all the insertions and deletions. Do not open that with my calendar. You're going to open it with Notepad. Hope you're not angry at me. That's not too bad. All right. So, whoa. Brian gifted 5 subs to us. Wow, thank you. That was far too generous. I don't even pay you to mod. That's very nice of you. Wait a minute. How close does that make us to subscriptions? Your coworker gave you money. All right, great. Well, thank you very much. Wow. Okay, 24 subscribers. Okay. I can do something. Why don't I just go ahead and gift like hold on. I got to do this. Gift. Hold on. There we go. Why not? Why not? Hey, now I'm second in the gifting thing. There, that officially puts us at over, I think over the tier to get more emote slots. So I guess more emotes to come now. Who did I actually gift them to? So congratulations to Passing Wind, Schmonite, Dracula, Samuel Animates, Montango, McFunk, Branrick Man, Adoinik, James Gurney, and Rinsunabe. There we go. Purchased ourselves some emote slots, practically. Okay. Anyways. Ooh, insertions and deletions. Uh-oh. Okay, so I know this looks pretty crazy right now. I mean, this is at least a little bit visual. This right here is giving a lot of information, most of which I don't even really know what's going on. But it's kind of saying, so for example, here, I believe that is an insertion. Yeah. So these are potentially mutations that maybe not I only have, but these are some of the bigger ones, some of the crazier ones. So there's some big ones here. So for example, let's go to one of these, and we can go explore any number of these. So we'll come back to CCR5. This is how we'll investigate CCR5. So we're going to go chromosome 1. Then we'll go to chromosome 1 at this position. Zoom out, zoom out, zoom out, zoom out. So it's not showing really right there, but apparently I have a pretty lengthy insertion right there. Okay, but I do wonder, so I'll have to, oh wow, there's a lot of information here. I'm gonna have to sort through these and actually like look at them in terms of not significance, but quality, whether or not it exists, whether or not I know. Given enough time and resources, could you read the whole code and figure out what it means? How far are we in that regard? I mean, that's effectively what a lot of scientists are doing, right? We are trying to understand even what things are doing when they're working. We have very little knowledge of what's going on when they're not working. So any particular mutation, you kind of just have to shrug and say, I don't know. All right, so quality, okay. So this is the problem. How do I go down to chromosome 3? Just, oh, there we go. So we gotta go back to CCR5 because I need the coordinates. CCR5 exists at, let's call it 46 million for, okay, I'm literally gonna type this in chat just so I remember. 350. There we go. Yes, so even despite the fact that we've sequenced the whole genome, details are still very much a mystery. I mean, here, look at this. I do actually have what might be a variant inside the CCR5 gene. We just don't know enough about what this T does to know if it's important, right? There's just too much information. There's 3 billion letters in the genome times 2 because you get one from your mom, one from your dad. It's kind of insane. And so each one of those billions of letters can be an A, T, C, or G, okay? And then all of those are, not all of them, but a subset of them are coded amino acid sequences. And so if you change the DNA, maybe it changes something. If you change the DNA, maybe it doesn't change something. If you change the DNA, maybe it changes an amino acid. Then you get into the next level of not just the DNA that's changed, it's actually the protein that's changed. And what is that doing? It's pretty crazy. But yes, it'll keep scientists like us in the job or with jobs for many years. Okay, I'm at 46 million. I've gone a scooch too far. Sorry. Okay. Seems like a job for AI. I mean, the problem is like, I mean, yes, people are trying to bring AI into it. We have these things called GWAS studies. I guess that the S stands for studies. It's Genome Wide Association Study. Actually, association or association study. One of those where basically we look at variants in many people and ask them every which question, like physical attributes, problems that they have, diseases that they've had, and we try to associate individual SNPs, or signal nucleotide polymorphism, those little changes that are different between human to human, and see whether or not they're associated with anything in particular. For example, that particular one that I pointed out before, that was the SNP which causes red hair. So there we go. All right. So I really wish I knew the exact position of the delta 30 whatever mutation. Delta, where are you? Delta 32 allele. If anyone knows off the top of their head exactly where it is. So this is a map of the allele frequency. So allele frequency meaning like the proportion of the population in these so a low proportion, but the populations that have this particular allele. They think it's Scandinavian in nature or in origin, so clearly some individual around here at some point had this deletion, and then their many ancestors have spread out over time. That is approximately where my ancestors are from, so that's why it doesn't guarantee me because, very small proportion. But, I wouldn't be surprised if I had it. Okay, I just got to find the where. Okay, this is all about where it is geographically. It's still super cool that a single SNP leads to red hair. Yeah, I mean, that's one of the easy ones. We can go look for other easy SNPs. Ooh, what about like male pattern baldness SNP? Let's see. Oh, nucleotide 794 to 825. Okay. Okay. Now, does that mean from the start of the gene body, start of the open reading frame? And how much do folks want for a decent interpretation? Decent interpretation of their genomes. So like you can buy interpretations of your genome. So actually, let me see if I can pull this up off to the side here. Actually, I'm not going to be able to now that I think about it. Where was this? So you can take these SNPs and run them through databases, and basically try and what they'll try to do is assign, like, oh, you have this SNP, this has been associated with this particular thing over here, this other SNP has been associated with this other thing over here, and so on. You're trying to find a better measure? Yeah. I mean, that's not 32 bases. I'm not homozygous for it, that's for sure. It looks like there's pretty equal coverage. I mean, that's not in the coding region, because it definitely said in the coding region. Although, like I said, I really don't, I kind of wonder. So back to this. So the CCR5 gene is basically from about there to beyond. Just kind of scoot it over. Let's see. 14,393. 14,393. 14. Whoops, too far. 14. Oh yeah, so the CCR5 gene is like contained within this. It does not look like. Nope. Thank you. Let's open this up. Also, the music is very, very quiet. There we go. So it's in here somewhere. Oh. Oh, hey, we can look for that. Yeah, you didn't. There's always music. It's usually louder for me. Okay, so we are looking for FPY SQY. FPY SQY. FPY. It's not a long gene. Okay. I'm just scanning. FPY SQY. FPY SQY. SQY. Oh, James, what? Yeah, you're welcome. Sorry, I'm busy. FPY SQY. Hold on. FPY SQY. Did I miss it? No, FPY SQY. Okay. FPY SQY. Oh, thank you. Thank you for typing it down for me. Yeah, James, we were, my mod Brian and I gifted 10 subs to everybody, honestly, to push us towards that next emote slot goal. So we're there finally. But, you know, want to give back to everyone. Yes, I am looking for HIV resistance. I actually don't think that this is going to show me because I kind of wonder if any reads span that, if they'll just be thrown out. Okay, wait. Wait, okay, I'm just going to put it over to the left. FPY. So, what was that? So it spanned, it was right after the FPY. And it spanned, okay, I don't know why. The deletion, okay. I see. So the, oops, get out of there. So the deletion should start about there. Do we need more? Do we need more subs? No, no, no. I mean, well, I'll always take more subs, but no, we've hit the goal. We've hit the goal that I don't have on screen. Okay, so this is why I'm going to have to do a little bit of actual bioinformatics off stream. Just because this is a fairly low count region. What? There's only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reads here. The chances of them being aligned in such a way that we'd see a deletion might be kind of small, because if there's a big deletion in or what appears to be a big deletion in a read, it might not be aligned correctly. Yeah, 30x covered my ass too, because this is not 30x coverage at this region or pretty much anywhere. It's not fair if they count the mitochondria, because that was like super, like hundreds x coverage, but that's because it's so tiny. Okay, so it's not looking good though for HIV resistance, to be completely honest. Yeah, although, no, that C doesn't mean anything. That's nothing. All right. Let's look at some other SNPs though. What's a good region to look at? Ooh, I mean, What is Huntington of Huntington's disease? What is that called? Huntington. I think that's how you spell it. Why did that pop up? Oh, HTT. Oof. Yikes. Okay. Huntington is huge. I don't know if I'd see an expansion. I don't know quite. COVID resistance? I could look at, hey, yeah, actually, Huntington is scary. Let's not look at Huntington. I'll look at that later. Maybe. Let's look at COVID resistance. So what is a COVID resistance gene? COVID resistance SNP. We found a couple, didn't we? Let's see. A couple weeks ago, we had found a couple because I had showed my previous DNA testing, but it wasn't specific enough to show all of them. All right, this is an article from Science. There is a GWAS database for humans. So, well, one of them is SNPpedia. It's kind of the less bioinformatic-y version. I guess, whoops, not COVID-10. Let's check this one out. See text on main RS. Yeah, see. Oh, actually, this might be the same one because I saw chromosome 3. All All right, having one copy of the SNP elevates a person's risk 2.6-fold, according to a preprint. Okay, but what is the actual mutation? Preprint. Give me your preprint. Okay, RS. So this is a different one. We will search this one. Let's see. I definitely searched COVID-19 and this did not come up. Okay, a higher susceptibility to respiratory failure. I kind of wanted to the happier one. So for Caucasians only, that's me. So I want a G or an AG. Okay. Okay. So the only problem is, how do I find where it is? SNP to genomic location. What if I want to identify genomic location by SNP? So this is just a name, right? And it gives me which like allele, A or G. But it doesn't tell me where it is. Okay, here we go. Query this. Yes, allow me to query. Why can't I? Sorry that I'm zooming around. Am I being dumb? No. Ugh. Okay, I guess I'm putting this in on another. Let's see, okay. I'm just entering something on this other side. HG19, my RS number is this one. So it actually has a couple. So they also mentioned this one. So I'll grab that because I can query multiple at the same time. Let's see if this just gives me what I want. All right, what I want is the, oh boy. Submit. Okay, this is frustrating. I want to actually look up what this is. Give me the location. SNP location. What are? Yeah, maybe if I just search it on NCBI. They gotta know. Ah, there we go. Okay. Perfect. Sweet, I can do this. Is HG19 the same as CH37? It is. Fantastic. All right. So that will be our answer. Give me the center line, please. Center line, center line. Can I not have a center line? whatever. All right, well, I've already forgotten what it was. 732. 732. I think it's A. There we go. I'm not congratulations to me. I'm not susceptible to bad COVID, basically. I do not have the high risk allele. Where can you find all the light commands? To be honest, I don't have an exhausted list. Does this work? I don't have an exhaustive list. Honestly, your best bet. Actually, is it even working still? Hold on, I'm gonna try something now. Color, red, green. Ooh, okay, my color thing might actually be broken. So light commands might be broken right now. I actually did, Brian, I did yours manually just now. Why is it broken? Something's broken. Okay, so there is a minor risk allele. Ooh. Yielded the strongest association signal across the genome for respiratory failure among patients. Okay. So this is not present on DNA chips used in the major direct consumers, blah, blah, blah. Ooh, okay. So I will search this one because it's not the proxy one, it's an actual one. So this is on this right here. So let's copy that. Check out this allele. Yeah, sorry, it's broken right now. You have to drive home and watch the rest later. Awesome. Thanks for coming, Megan. It looks like I'm good on this one too, actually. Yikes. Okay. So I need more alleles. I need more alleles to look for. I literally have my entire genome. The problem is, like, where do we even start? I mean, look at all this stuff. So I could, okay. Maybe I should open this in Excel, actually. Excel is not going to be happy. Let's also look at the SNPs. Let's open that one in Excel first. Open, where's open with? Ugh. Gonna have to open Excel separately. I might be able to actually sort things by like confidence or something like that. So let's open this up on my other screen, of course. You're going to open all files and you're going to like it. Import at row. Jeez, okay. Skip like the first 100 rows. Okay. 44. Ooh. Actually, this is not going to work. Or maybe it will work. Please don't hate me, Excel. Please don't crash everything. You always use R for this type of sorting. Yeah, I would normally. I just was not mentally prepared to use R tonight. But you're absolutely right. The best way to do this is R. And also, I normally look at VCF files, so that's what's most useful. probably at the moment. Uh-oh. That was the sound of my computer not liking it. Yeah, that's not going to be useful. Oh well, I tried. Yeah, maybe next time I explore this, I will definitely be using something like R. TP53, TP53. So TP53 I know is, it's an oncogene, right? So what is a good TP53 allele to look at? Let's not look at that. Actually, SNP is probably what I should do. SNP. So SNPs is prognostic and predictive factors in patients with breast cancer. I do wish there was like a like happy SNPs we should look at, because I don't want to get sad by finding out I'm at risk for a particular thing. Even though, to be completely honest, everyone is at risk for, not everything. Everyone is at risk for something, and probably also has mutations that get them, give them protection against other things. So it's a give and take no matter what. You know, I'll at some point I will find alleles that's like, oh, this is not good for this type of cancer. Well, if I have that, then you probably have some other, here we go. You probably have some other series of SNPs that make you susceptible to some other type of cancer. Okay. Ooh, 1 of 25 SNPs reported to represent an independently minor but cumulatively significant increased risk for breast cancer. Okay. Cool. Okay. So there's quite a few. Interesting. So this one is already, I assume this summary, live 3 years longer, chemotherapy is more effective. I assume that's in the context of having breast cancer. So let's actually look, oh, it gives the position right here. Wow. Oh, that's for the wrong reference though. I should definitely make sure that I'm looking at the right reference. Okay. So 472. Oops. Chromosome 17, 472. Oh. Okay then. I appear to have, whoops, something. What do I have? Oh, that's a good one. So I'm homozygous for the CC genotype, which is live 3 years longer, chemotherapy is more effective. But again, I mean, it is genetics, so that doesn't actually mean anything super significant, but it is associated with that. Okay, I'll take it. I'll take that. That's a good one. That's it. See, good SNPs. Only good SNPs this stream. All right, let's, what's another one? So we're at, this is 522, 2222. So they mentioned a whole bunch of them. So it turns out, let's see, CEU. So this is Utah residents, but northern and western ancestry. So I actually have the sort of minority allele for my general ancestries. That's cool. All right, I'll take that. Let's look at another one. But see, the next one I look at could be the opposite, so it might just even out. I should look at the BRCA1, BRCA2. So TP53. Okay, I gotta get that full text. Okay. So what are we looking at here? Genotypes in 473 cases. Yep. I would just like these SNPs, please. Individual SNPs carried by each of the 21 cases. Okay. But I would really like the SNPs. Just specifically the SNPs. So those are the specific mutations. This is kind of a strange... Okay. I was just kind of hoping for a just a table that would just give me the SNPs. Okay, if you are going to... Okay. For details, check the breast cancer entry. There we go. Ah, okay. Oh, wow. Okay, we can... There's a lot of them. Okay, so I have the good TP53 gene. Oh, interesting. How to calculate the estimated risk. It provides two different algorithms. The first is to simply count the number of risk alleles for these 25 SNPs, i.e. the number of rare alleles for each SNP. A homozygote for the rare allele ups the count by 2. Okay. Well, we're just going to do back of the napkin. Ooh. SNPs that are associated for lower risk of breast cancer. Okay, let's do that one. Those two, because there's two and lower risk. I want good SNPs only. Okay, so I got to search you because my genome is currently mapped to an annoying version of the human genome. Okay, chromosome 7 at 07. Oh, uh-oh. Well, I'm 50-50. So I've got the T. Well, hold on now. What? Excuse me? Oh, did they send me the raw reads too? Yeah, they absolutely did. So they said this is their aligned reads though. I'm just gonna have to deal with that until I get the chance to actually take my raw reads and align them. I'm very curious because they said it should be 30x coverage, and this doesn't look like 30x coverage necessarily. At least it wouldn't if it was the organism that I study. So maybe I can complain to them and get them to sequence more. I doubt it, but who knows what they'll do. But yeah, and then also I want to more carefully look for insertions and deletions and mark them because I have no idea what their parameters were when they aligned these. Base G. Okay, this is the reverse. Okay, great. Whew. Show all bases? There we go. So CT, yeah. So I have the lower risk for breast ovarian cancer. Nice, that's 1. Do I get double though? Whoops. So that is in the DNA repair gene. Cool. Next one is caspase 8. I think caspases are involved in apoptosis. Let's see. So this one is... And yeah, so they also sent me just VCF. So honestly, what I should be doing is paying like the $10 that it costs just to take all of this information and put it through a like pre-made pipeline, analysis pipeline. Although I guess I could do that on R right now. I did not think of that because I'm being dumb. There's got to be some. Actually, James, so you've mentioned already GWAS for humans, I think you said, right? Does that mean you know of GWAS databases for non-humans? As in you might know how to query just wholesale, um, just like look for general risk factors or things like that? Do you know of any R packages that do that? All right, I'm looking for, what did I just say? 89, 89, 89. Looks like I have the normal allele for that one. That's fine. Your wife does GWAS, not you. Ah, I see. Well, let's just search that quick. I mean, there's got to be not RStudio, R package, GWAS, or SNP risk actually. SNP risk. Sambar. An analyses of biallelic SNP datasets. All right. Import data. Import a SNP from bio. Oh, well, I don't have that. What if I do VCF? So yeah, a variant annotation, annotate variants, compute amino acid coding changes, predict coding outcomes. That could be good. Yeah, I might have to do this later. I think it'd be pretty boring to watch someone do that on stream, so I'm not going to do that right now, but maybe I'll try to do that in my free time. Variant annotation. Let's see if they have a vignette. How is the okay, now you have started your new position, right? How is that going? Let's see. Ooh, possibly damaging, benign. Okay, cool. I think this is what I want. It's just more text. I love a thing that gives out a graph, but don't think I want to do that. So the thing that I'm thinking of is Prometheus. Whoa, Prometheus. So Prometheus is kind of cool. I agree to all these. Check. They're basically telling you on this that like, hey, this is not medical advice. So let's do a sample report. So what these reports do is they take these SNP tests basically and looks around, skip, and it kind of gives you a summary. So, and you can kind of do it by good, by bad. It just kind of summarizes everything. So like, for example, this example, the thing that they're most sure of, it's genetic female, but then, hey, also 2x risk of Alzheimer's disease. Also eye color, age-related tasting variation. Whoa, that's interesting. Actually, I want to look at this one. So these are all based on GWAS studies. And also, yeah, haplogroups, so that's kind of cool. Now, I've done this before with my Ancestry DNA results, but that is not as complete as what I have now, which is every variant, basically. But there would have to be a way that I curate it to also grab all the ones that they expect to see. Oh, it's going well? You're still trying to order stuff? Yep. Writing grants and get your PhD student hired. First student already. That's awesome. But also grants. Like, yikes. That's not fun. Okay. So let's see. GS227. SNPs known to influence the ability to taste bitterness. This means you're better at than average at detecting bitter tastes while young, but that ability decreases to less than average during adulthood. As a child, you will probably hate Brussels sprouts. In early adulthood, you will probably discover that olives and Brussels sprouts now taste good. I want to look up this one for myself because I used to hate things like olives. And now I kind of like them a lot. Same thing with beer as I got older. I hated it at first, but then I started to kind of like it. So I'm going to open all three of these actually. And they're all in the same gene. That's kind of interesting. So let's see. Oh, you're the same way with mushrooms. You still hate the taste of beer, though. Some mushrooms just taste like dirt to me, but also it's just the texture. I don't like the texture of mushrooms. Okay, so do I want... What mega receptor is this? Yeah, it's the TASR38. I'm guessing the R stands for receptor. Let's do some... let's search this. And then, so TAS2R? There's a bunch of them. What was it? 38. TAS2R38. TAS2R38. Whoa. Wait, what's... Oh, all right. I'm looking at all the DNA. Right. Shh, don't show all bases. Okay. You apparently like the favorite dirt because you're... Or like the taste of dirt because your favorite tea is puer, which a lot of people say tastes like the forest floor. So I've had that. I like it. I think it has like an earthy taste, but not like dirt. Definitely. I see. Okay. Well, already it looks like I have something right here. So, oh God, you've started a contentious thing. Well, I'll just say I love it. Since apparently there's no in between. Okay, so this one. Which one of these alleles is it? Okay, so it's taste2 receptor membrane 38. Wow. Cool. Hold on, I saw a longevity. What? Okay, so let's read about this gene. This gene encodes a 7-transmembrane G protein coupled receptor that controls the ability to taste glucosinolates, a family of bitter tasting compounds found in plants of the Brassica species. Synthetic compounds, PTC and prop have been identified as ligands for this receptor and have been used to test the genetic diversity of this gene. Although several allelic forms of this gene have been identified worldwide, there are two predominant common forms, taster and non-taster, found outside of Africa. These alleles differ at 3 nucleotide positions, resulting in amino acid changes in the protein. So A49 to P, A262 to V, and V296 to I. So AAV. So it looks like this one might be one of those, actually. So I think that's the one I have, Topaz Puer, because that sounds really, really familiar. Ooh. Do I have a second? Do I have a third? Hold on. That's an I. Wait, was it an I or a V? Okay, now these are fairly low, so those are probably not real. I think I have one. I have one of these. So A49, so that one was probably A262. Yeah. So I only have one of them. Okay, let's go back and actually look at them though. Fortunately, I gotta look these up. Nope. That is a definite no on that one. So that was 39. So let's go to this one. Search this one. Okay, that is the one that I have. So possible, unable to taste bitter. All right. Oh. You're giving us more instructions. You can re-steep the same leaves like 4 or 5 times, so it's a good value too. Oh wow, okay. Maybe I should go dig that out of my closet and actually out of my cupboard and actually use it. That sounds cool. You've never really ventured outside of the basics. You mostly drink chai tea, but you generally love hot drinks. LOL. I wonder if there's a allele that makes you like pumpkin spice lattes, because I absolutely love pumpkin spice lattes. I have not had one yet, but it is pumpkin spice latte season in the US. I think they taste far too sugary, but I do like the taste. Okay, I got that one too. But I can still taste membranes. Yeah, well, it's a texture thing. I have a textural problem with a lot of foods. All right, so I have one of the 3 alleles, so not sure what that means. That was a fun little digression, though. What else is on here? See, I can just look at this. I don't want to look at that. Let's look at good. Let's sort by good. Blood. Oh, I can double check my blood type. Yeah, this is awesome. You have food texture issues too. You're almost an accidental vegan because meat is a minefield of potential problem textures. See, for me, it's fruits and vegetables. Certain fruits and vegetables have a particular texture that I don't like. Like onions are the big vegetable ones. Honestly, it's mostly just onions. And then some fruits are a little like, ugh. You're definitely going to sequence your genome after this? Well, do it. I don't know. I feel like you do have to be a little bit brave because, there is actually the serious risk of uncovering something sort of scary. But if you've so I had already done a little bit of searching through Ancestry. So I already had the major jump where I was like, okay, there's nothing super clearly wrong with me at the moment. However, wait, what the heck? There's something clearly not wrong with me. So I was okay jumping in for the full thing. Is this a little tiny intron? I don't understand. That's too small. Oh, that's probably an entry site or something. I don't know my intron sequences. So this intron right here is probably skipped. Not like it goes from V to T. What was I looking at? 09. So I have the T, which I believe is not the D. So yeah, I think that's it. Yeah, it's too small for an intron, but I think it's like an alternative splice site, actually. I bet you that's what it is. All right, let's look at the next one though. I do know my blood type, but I kind of forgot it. I could look it up. I could look up my blood type. What did I just do? Okay, 322 cc. Oh, wait. Shoot. Oh, no. Actually, wait. Never mind. Never mind. There's actually way more. There's actually way more things that contribute to blood type. Maybe I won't figure that out. I do just want to go back. I want good. Repute. Give me only good, please. So these are not my results, but these will help me. Ooh. Extra tasting ability. Rare T allele. Better at detecting umami taste. Okay, see, that's something I'll want to look up. Do I taste umami? I do really like savory things. I prefer savory over sweet, definitely. But I don't know if that's because I can detect them better. Chromosome 1. Oh, I have an allele. It is probably not that allele, though. So, where'd that go? Where'd that go? Common and complete genomics. Oh, well. No extra tasting ability. Sad. For women, 60% reduction of HIV viral load. Definitely check that one out. So this is a SNP in the 5' region of the HLA-C gene. 35 kb away from the transcription initiation. Wow. People with this tiny sequence variation appear to have 90% less virus in their systems than those who carry other polymorphism. Hello, David. Welcome back. I've just been searching around some more SNPs for myself. I unfortunately don't taste umami very well, and I can taste a little bit of bitter. Things I absolutely would not have known if I hadn't have gotten my entire genome sequenced and checked it out bit by bit manually. Go. Huh. Well, 2 reads, 50-50. What does that mean I am? 60% reduction in HIV viral load. Potentially. Hey, I'll take that. I'll take that very much. What is the... Cool. So that doesn't necessarily mean anything, but that's still pretty cool. Potentially. Oh, the cilantro thing. Hey, that's the good one. Cilantro SNP. Yes. This is what I've been wondering this because I think cilantro does taste like soap, but I still like it. Okay, we find a single polynucleotide or single nucleotide polymorphism significantly associated with soapy taste detection that is confirmed in the cilantro preference group. Okay, so let's search this one. You have the cilantro thing. You can't eat it and can detect even small amounts of it. I've actually been wondering this for years. I forgot about this one. Okay. Least likely to think cilantro tastes like soap. Okay. Cool. There's actually not a great summary for this one. Oh, there's a proxy allele. We'll look at the proxy allele as well. So this was not included in my genetic results, my ancestry DNA results. Whoops. Oh, yeah. Okay, not great coverage. Not at all great coverage. But I am not a variant. What was that number again? 48, 48. So it looks like it is G. So, wait, more likely to think cilantro tastes like soap? So that's the thing. I do think it tastes like soap, but I like it. I like the soap. Okay. So there's a proxy allele. So let's check this one out as well. Whoops. Okay, so I have one soap allele. How many soap alleles do I have, though? I think this one was also a G. I've already forgotten it. 26, 26. T. More likely to think cilantro tastes like soap. All right, so yeah, how did they build this as 30x? I do not know. So I actually don't know the coverage for sure. We'll zoom out and kind of look around, poke around a little bit in the last like now 15 minutes of stream. But I need to figure that out. I actually don't know how you can. I mean, I do know how you can calculate coverage. I do not know of a simple, easy tool or even like thing that IGV does to just calculate coverage quick. Just an estimate. It should get more attention. It matters when you can say it's genetic. I'm not just a picky eater, Dan. Well, okay. I see you, Managard. I respect your opinions, but it appears that I have genetic evidence that I do think it tastes like soap, and yet I love cilantro. I love it. Absolutely love cilantro. Like when I can throw cilantro into something, I do it. So maybe it is just a preference thing too. Like preference must have something a little bit to do with it. There's probably more to it than just these two, but they seem like they're good predictors. You are not a soap taste fan. Not to say I go around like, chugging Dial soap just to get my soap taste fix. All right. Zoom. Okay. Oh. Oh, that's the other allele. I've got a couple. Whoopsies. What was the name of that gene? Or 6A2. Or 6A2. Was I looking in the wrong spot? I saw it go by. Also, it didn't work. I feel like chat commands aren't working. Chat commands are not working, and I can't figure out why. Hold on a second. Hold on. Something's broken with Leo Ranboard. Twitch chat. I think it's not connected. aren't you connected? The main Twitch chat account has expired. Oh, my token has expired. Oh. How do I get a new token? Twitch chat token, please? I might have to do this later. Yeah, I'm gonna have to do this later. Yeah, sorry everybody. I didn't realize that it was didn't realize that something was going wrong. You've refunded your own points. Got it. All right. Why do I have so many variant like alleles in this particular area? And this was one of the two, right? This G, or was I looking in the wrong place this whole time? What is that? 78, 1178. Let's go back. Huh. Very, very strange. What is this allele? Huh. I'm looking down here at other alleles, I believe. Okay. I feel like there's something going on there. I don't know what. I'm homozygous for something. All right. So now I'm zoomed way out. Yeah, see, there's a lot of variation that I wasn't really expecting. What are more good genes? What's like, I don't want to look at anything scary. It's too late for that. Much lower, 0.15x for type 1 diabetes. Sure. Let's look at type 1 diabetes. So this is an HLA gene. I've got a bunch of A's, but it looks like I'm heterozygote. I have a lower risk. I don't know why much lower is 0.15 and lower is point. Oh, I see. Okay. Cool. Well, that's good. All right. Does anyone have? Oh, another. Oh, I have a. What? Why? I have so much variation. That's cool. Am I supposed to have all this variation near all these HLA genes? Probably. It's kind of what they do. So now I'm just zooming around. I want to know what's another gene? How about the gene? How about the gene that I studied in grad school, which is big? I've got some variants here and there, but nothing in an exon that I saw. That one, no. That one, no. Ooh. Ooh, maybe. Splice isoform. Oh, no. Not at all. Okay. So I've got all these. So I've got all this data. Heading out, David? Well, thanks for coming. I'll be closing up shop here soon anyways. Have a good night. I'm still really interested in these insertions and deletions, though. Especially, like, I just don't know how it's... How are they determining this? I don't do enough with VCF files. Like, that doesn't show me a huge insertion. This is a big deletion right here, but we're near the telomere here, so it's super repetitive. Or at least it should be. A lot of weird stuff going on there. I just want, like, what are these huge insertions? Let's see. Del? Is there just del? Oh, okay. Am I looking at the right columns? So we've got ID reference alternative. So the alternative is what I have. Better double check that. So where there should be an AC, so I should be able to see the difference here. Where there should be an AC, I should have 1. So there should be a deletion. Ooh, yikes. Yes. So that is a deletion of 1. Okay. Ew, in this gross telomere region. That is, what is that? Is that super GC rich? GT rich? Or CA rich? It's really CG rich. It's not just C's and G's on the same strand, it's both strands. Okay. Okay, so that is, I am reading that the way I think. So where do I have, I see. So like this one is a big deletion. Okay. Reference alt. So we were looking at the right thing if we were going to find something for that CCR5 gene for HIV resistance. I do not think I have that, unfortunately. A little sad, but that's okay. All right. Let me go back to Prometheus. So I wonder if, so my filtered SNPs. Oh. Hold on. My cat wants to escape. She was very mad at me for locking her hair in. All right, so I somehow have to turn this on that side into, gotta find it, on my PC. Okay. So I need to turn what's on the left hand or the right hand side into what's on the left hand side. The difference is pretty extensive. I don't know how to do a line count in just, you can't, I don't think you can do it. Let's do Control F, RS. I won't give it to me. Still, if I can turn the left hand side or the right hand side into the left hand side, I should have a record for every single known SNP in the human genome, which I can then put into Prometheus to get a personal DNA report. Upload raw data. All right. Let's see. And I should open this on the other side so people don't see my login information. But I should actually just go straight to the company that gave me these results. Okay, we're just gonna quickly change the window size so you don't see my personal information. At least while I'm setting this up. All right, so these people, they sell reports. I know that just made it like super small, but it's more wide than anything else. They sell reports, but they give certain reports. So let's actually look at one of these. Okay. Blah, blah, blah. So the only annoying thing with, so this is effectively what I could get done. Oh, you have a pipeline for R that does code for, that does this for bacteria from VCFs. You just convert the VCF to CSVs, then they do an inner join. Yeah, I basically just need to take the time to pull up the correct databases, and then yeah, all I'll have to do is like join them. Although, let's see here. I just need to make sure I have the position for all of these. But the thing is, so this SNP, I don't exactly know how they do it. I don't know if all these positions are necessarily recorded, because I want the ones that are even, like they are not different from the reference. And I believe these do not give anything if they are not different from the reference. If that makes sense. If what I just said made any amount of sense. So let's see. 8.2. Oh, these might be different genomes too. Reference genomes. Yes, I'm not seeing that one. 7.2, 7.5. Okay, that is one. And it's the same. Okay. So maybe it is? I don't know. I have to check. Well, there's a lot of information in these. Yeah, but you're right. I absolutely can do this. I just have not had the time to do it. But I would love to get more information, because like this company that I went through, you know, they give these reports and you can buy more, which you should probably not do that. But they don't actually give much information. I don't think they do. So let's see. So they flag some results that indicate my blood pressure would highly benefit from a lifestyle with plenty of physical activity accompanied by a proper and healthy diet. Well, they don't say why, like what that is from. And, you know, I'm sure there's some GWAS data, some single study that showed that, but they don't link to it all. They don't cite their sources. Ooh, enhanced. Enhanced. Your genome suggests you are predisposed to have more extraordinary sprint, meaning you will benefit from cardio fitness such as track and field. Yeah, that is not me whatsoever. Let's see. Your genome suggests that you are predisposed for better physical performance related to speed. Yeah, not if you've seen me try to run. I am very slow and very bad at running. I'm predisposed to more benefits from resistance training. Ooh, it is advisable to train 2 to 3 times a week. Ooh, okay. Yikes. Oh, hello, Elmbis. Welcome. We're reading up on all my genetic information. Injuries. This is a long report. 45 pages. Whoa, anonymous gifter. An anonymous gifter? Oh, Fossbotch is screwing up. Gifted Embis a sub. Thank you, anonymous gifter. Some well-deserved anonymous gifting to Embis, who, from my recollection, has gifted other people like 10 subs, maybe 15, something crazy like that. I apparently have strong bones. I've never broken a bone. All right, let's see. Metabolism. Ooh, low metabolism. I only want the good stuff. Wait, what? Oh, they do. They do actually give. Okay. So if I search bones. Okay. So these reports actually do give more information. That's good. I was a little worried. Yeah, yay, strong bones. Yeah. I mean, I'll take the strong bones. What else do we have here? Nutrigenetic report. All right, let's check this out. Ooh, fancy. So I can buy a ton more. Uh-oh. Increase the intake of fishy oil to decrease the consumption of vegetable oils, which are too. Ooh. Well, probably care. Let's see. Circadian rhythm and appetite regulation. You should try to educate your body to have very regular rhythms. I do this. I'm increased risk. Oh, increase my fiber intake. And avoid simple sugars as much as possible. Interesting. Oh, none of these are great, but I have to read more into the report, I guess. Caffeine. Oh, caffeine sensitivity. This is not going to be good. Opt for caffeine-free drinks and herbal teas. And avoid excess dark chocolate and pure cocoa products. Ooh. Okay, I'm going to have to look at what they say for this one. I'm going to Control F that because I drink a lot of coffee and I love it. And I also like dark chocolate. So, yeah, that's a nope. I don't, yeah, exactly. All Mr. I agree. I don't care what my genes are saying. Carriers of the C allele may experience anxiety after consuming products containing caffeine. Very true, but I still love caffeine. Especially if they're infrequent caffeine users. Therefore, I'm fine because I'm a frequent caffeine user. It's more marked in homozygous carriers, people suffering from panic and anxiety disorders. Carrying the risk allele should abstain. Ooh, yikes. Yikes. This is a case of shut up genes, you're not my dad, but they came from my dad. This is feeling like genetic astrology, everything's very generic. I mean, Embus, you're not wrong. I mean, this is right. I have all this information, and also scientists with a laser. Welcome. Yes, I did. I have all this information, but like, how much of it does it actually determine versus and tell me? That's kind of the thing that's up in the air. It's more out of like pure interest for me, to have this done. So it does feel a little, especially these reports, very genetic astrology. Yes, I did get my DNA sequenced. I did not get, well, I actually have here. Let's go to there and there. So scientists with a laser. So on the left-hand side, I have my ancestry DNA results from several years ago, before 2014. And on the right here, now these are just the SNPs that I got from my whole genome sequencing that I just got back yesterday. The company I went for is called Dante Labs. They were having a back to school sale for $200, and I got it back in like three weeks, which was half the turnaround time that they said. However, if you know anything about DNA sequencing, what they promised was not exactly what I got back. So I'm going to have to do a little bit of digging myself. I'm going to take the raw data and realign it. And we're going to actually see if I can't like complain and get not my money back, but maybe more sequencing done so I get higher depth. I also need to learn more about the human genome. I know a lot about Drosophila, like fruit fly genomes, but not the human genome. Gotta learn. Yeah, so it's called Dante Labs. Oh, you're writing scholarship applications. Ooh, well, good luck with that. Um, you mean scholarships or grants or something in that area, that genre? Good luck with that, regardless. I am looking for what I was promised. They said 30x, and they should have expected at least a scientist or two was going to use their services. Oh, my window's still super small. That's because I don't want you to see quite everything. All right, I'm going to go back up to the top. Alcohol sensitivity, normal. Okay. Let's do the last one. So I did fitness. Oh yeah, so you can buy the rest of the reports. Here, let's shop reports, which I'm absolutely not going to purchase because it's pointless. Health, circadian rhythm. Oh, see, Embus, these are all the astrologies that you can get. You can look at your endocrinology report, hematology, blah, 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 blah. I mean, the data's all there. They just use their proprietary, probably not even like, they're secret within the companies, but they're probably using fairly publicly available data sets to figure all this out. Uh, Sorry you missed the part where I talked about my data, but yeah, I do have a bunch of information. Yeah, I'm super excited. I'm going to stop looking at that. I'm just going to go back to this so I can re-reveal everything. There we go. What I would actually really love to do is take all these reads, look at these variants, right? Use the calls for the like high quality variants, probably from those SNPs and also those insertions and deletions. I want to make my genome, like my just A, T, C's, and G's, all 3 billion letters, except for the variant sites where it will have to be a different letter to denote the two alleles. And I want to do something with that, like artistic, but I don't know what. I've already calculated, like, okay, what if I wrote it all down in a book where every single base was like a single dot in like a 300 dpi image, which is pretty small. It's like the smallest that most people can distinguish on a page. but I did the calculations, and if I did that on an 8.5 by 11-inch page, front and back, and made a book out of it, it would be 100,000 pages long, which is a bit much. So I don't think I can do that. Although we also did say, I took a vote and everyone on stream said, hey, I should just publish that 100,000 page book as a book on Amazon. So maybe I will look into that. I got to write a program to like generate that though. And you're applying for funding for your PhD. Well, good luck. Finding funding is never fun. But hope you get it. Let's see. We looked at Prometheus. Yeah, so I'm going to have to work on Prometheus. Best SNPs to have. Best SNPs to have genes. I don't suppose there's a top 10 SNPs. I already searched this. Top 10 SNPs to check. Huh. Here we go. You found out you can apply for grants to get funding to benefit low-income students from the state. You really need lab supplies, so you hope you can find something you can get. Yeah, I hope so too. Yeah, I remember my high school teacher managed to get us a grant, and like she was able to buy some like really cool lab supplies with that. Oh, okay. Ooh, Iron Overlord. Overload. We were talking about this with about vampires. So hey, let's see actually if I would be a successful vampire. So hemochromatosis caused by an increased iron storage in various tissues in the body. Yeah, let's look up this one. I want this one. All right. I'm ready to serve my vampire overlord. All right, what did I just type? 41. So I have the G or C allele. Probably should double check that. Oh, typical. Oh wait, no, that's good. That's good. I am not susceptible to hemochromatosis. Nice. Adam 12 might have genius alleles? What? Adam 12. The American crime drama from 1968. Intelligence. Oh, there's so many. Brian, did you have any more information on that? I mean, I'll look at Adam 12. Wait, Managard, you actually knew what that show was? I've never heard of it. Why can't the good things be on Simple G? What? Okay. Hot take. Mammalian genes have too big of introns. Like, what's the point? Why would you bother? Actually, introns are super important. They're super important places to put regulatory information. But still. All right, I have an allele here. Introns are the worst part of RNA. Okay, DNA is going this way. right? GTG, yeah. I mean, I have an allele here. I guess Adam 12. I don't know if this will. Oh, thank you. Thank you very much. So let's look up these ones. I think I'm going to look these up and then I probably should go to bed because I have teaching tomorrow. That one doesn't even have an entry. Let's see. So that one was 65. I have the standard allele for that one. It's supposed to be a C, so I guess I have this backwards. Yeah, okay. All right, next up. Yes, I do. Yes, exactly. I need to train my body in circadian rhythms. I am usually pretty good about going to bed right on time. This one also, no information. Hey, if there's a paper based on that, you should let me know. But no big deal. I can just search it later. Oh, shoot. And I saw your reminder for a raid, so thank you. Yeah, so as I'm searching this last couple alleles, with no idea about whether or not it's important, the general plan is on Sunday. Sunday, apparently, we're doing a card opening stream. Not probably the whole time, but I'll get as many packs of cards as I feel like is reasonable. Packs of cards. I'll try to make it very official. I'll get a camera set up so that we can watch them being opened together. And then next Wednesday, I'm really hoping to have on someone to come talk to us about chromosomes, chromosome pairing, and how we're going to try and sort of scientifically not rap, like singing, like discuss, hypothesize how different alien species could breed, because there's a problem when you try to pair up your chromosomes as a alien species with another alien species. All right, so I have one of these alleles. What does that mean, though? Anyways, new Pokemon cards just released. Hey, I'll grab some of those. Yeah, it's gonna be really fun. I think it's gonna be less because she is. I think she has some background in chromosome biology, but I believe these days it's more of a very strong interest in that and science fiction. So we're gonna have a good discussion, I think. Absolutely nothing, but you're still a smarty. Oh, thank you. I'm smart because this is a C, is what I will say. Although it's in an intron, so I wonder what that does. Oh, I will definitely copy this. Oh, yes. I got this. Don't tell what's his face, Joffrey Miller, about this high intelligence. Okay, so who should we branch? Yeah, branch site shit. Who knows? Is it an A? Oh, it is an A. Yeah, who knows? We'll see. Okay, so we should rate someone. You want to have your whole genetic code now? Well, it's here. Don't look at that. Let me DanteLabs.com. Here, I might as well. DanteLabs.com. Also, sorry for those of you who have shoutouts. They haven't been working today for a reason that I know but didn't have time to fix. So, yeah. Anyways, let's go raid someone. So I saw, Brian, you reminded me. Other Marcus's rating is on. Yeah, sounds like he's doing a couple Wednesdays. Science streams and freckled science. Ooh. Oh yeah, you can promo everybody. Let's see. Gotta get my ducks in a row here. Oh, you're really going for it. All right, so thank you for everyone for coming, lurking, chatting, or otherwise. Same thing with gifted subs, whether we do know who you are or we don't know who you are. Let us raid. Freckled science. Let's see. I don't know what they normally talk about, but let's give her a try. All right. Let's go to big cam. All right. Let's see. Gotta check up with the chat now because I looked away. Yeah. Thank you everyone for coming. Scientist with a laser. Hey, no problem. Thanks for coming. I appreciate you dropping in, even if it is late. Bye everyone. See you later. Stick around for the raid. Otherwise, I will see you Sunday for cards and microscopes. So that should be fun. Okay. Bye.